Hi guys, today we're going to talk about the 3 stages of cell signaling. And then we're going to talk a little bit about type 1 and type 2 diabetes. And then the protein versus steroid-based pathways we'll do together in class. So, like I said, this unit's a little different than other units that we've done because this is something that you typically haven't learned much about before. And so when we talk about the 3 stages of signaling, the first stage is going to be reception, and that's going to involve a signaling molecule like a hormone binding to a receptor. And that receptor can either be located in the cell membrane or it can be located in the cytoplasm, depending on what kind of molecule this signaling molecule is. Okay, then you have transduction. And basically all that means is it's a series of steps typically taking place in the cytoplasm, and it's a way of getting whatever message this signaling molecule is trying to send. It's trying to get that message deeper into the cell, usually to the nucleus, to bring about a cellular response. In other words, maybe we're trying to tell a certain gene that it's about to be transcribed. That might be the cellular response, and that often is the cellular response. Okay, so let's go through what happens in each of these stages. Now, really today, we're just doing an overview on these 3 stages because what we're going to learn about over the next few days are specific pathways. We are doing the steroid hormone pathway today, which is pretty easy and straightforward. But then either Thursday and Friday or Friday and Monday, you're doing the G protein pathway and the receptor tyrosine kinase pathway, which are more detailed, so you kind of have to have an overview of what's going on there. So in the reception stage, the chemical signal is going to bind to a receptor protein, and like we said before, that protein can either be on the cell's surface, like on the cell membrane, or it can be in the cytoplasm, and that's going to depend on whether our signaling molecule, our hormone, is protein-based or lipid-based. Transduction. So keep in mind, remember we talked about receptor proteins in the cell membrane in the last unit, and we said that they're not actually trying to get a whole molecule into the cell. Like if we go back to this picture, we're not trying to get this signaling molecule into the cell. What we're trying to do is send a message to the interior of the cell. Okay, so the binding of the signaling molecule to the receptor protein is typically going to change the shape of the receptor protein and allow something else to happen. Maybe some other molecule comes in and binds to it. I know this seems kind of vague and abstract today, but trust me, when you learn about the specific pathways over the next few days, you'll say, oh, okay, that kind of makes sense now that we're doing specifics. All right, B. The signal transduction pathway usually involves several steps. We're trying to get the message from the receptor protein deeper into the cell, usually to the nucleus, so that we can activate a certain protein to be made. Okay, so each step typically changes the next molecule in the pathway. Some of these pathways involve cascades. It involves one thing activating the next thing, which activates the next thing. Okay, and that can activate enzymes in the pathway. So what we're going to talk about a lot here is what happens when one thing goes wrong somewhere in this pathway. How does that influence the rest of the pathway? Okay, then we have response. Let's just take this a little bit at a time. Response just involves the specific cellular response. Like I said, a lot of times this is going to involve activating a certain protein inside the cell, and that protein can go bind to a specific section of DNA and cause it to come undone, cause it to come unwound, and activate that section of DNA. So maybe in biology honors, maybe you heard about the promoter. This protein can bind to the promoter, which is like a regulatory sequence of the gene, and it will signal to that gene that it's about to be transcribed. It's about to be made from DNA into RNA. So a couple things can happen with the response. It could cause an enzyme to be produced that breaks down a given substance inside the cell. So the pathway that we're going to talk about on Friday is the G protein pathway. And the final step in that is making an enzyme that can break down glycogen into glucose, and then that glucose can serve as fuel for your cells in the case of an emergency. Or it could be, like we said before, a protein binding to a specific section of DNA. Remember, when you replicate your DNA, you're replicating an entire chromosome. But when you do transcription, you are only transcribing one gene at a time. That's kind of like turning on light switches in your house. We're only going to turn on light switches for the rooms that we're in right then. So we're not going to go ahead and make all of our DNA into RNA. We're just going to make DNA into RNA for the genes that we want to make a protein from. You might be activating a protein to let it go bind to a specific sequence of DNA to tell it that it's about to be transcribed, it's about to be made into messenger RNA, and then that can eventually be made into a protein. Okay, so we're going to go over this pathway a little bit later, but what I was meaning by that is, in this case, this is a steroid hormone pathway, and notice that this steroid receptor complex comes into the nucleus and it binds to the regulatory site, probably the promoter of the gene, and it basically tells this gene, hey, we're going to open you up, we're going to break the hydrogen bonds, and we are going to make mRNA from you. So here's that mRNA strand, and then that mRNA strand can leave the nucleus and come out to a ribosome and be made into a protein. So when we say something bonding to the DNA, this is what that would look like. Okay, couple things about receptors and their signaling molecules. Receptors are specific to a given substance, and then hormones can only act on specific cells. So in this picture, you have these different molecules out here, and they don't have a receptor on this cell to bind to. So they must not be able to act on that cell. But this molecule, whatever that may be, does have receptors on this cell, which means this signaling molecule is supposed to act on that cell and have its influence on that cell. So it says, although epinephrine is your adrenaline, okay? We'll talk about that more on Friday. Although it encounters many different cells on its journey throughout the bloodstream, only certain target cells will have receptors for this hormone and can react to it. So think about when you get excited, when you get ready for an emergency, you know, your heart rate speeds up, your bronchi, your airways of your lungs are going to dilate. Those cells would have receptors for epinephrine, where like your bone cells probably don't. They're not really activated by epinephrine. Okay, ligand. You're going to hear this term a lot in this unit. A ligand is a special name for a molecule that binds to the receptor. So here, a ligand is binding to a receptor, and then it's going to cause something else to happen within that cell. So usually when a ligand binds to its receptor, it causes the receptor to change shape in some way. That's usually part of the process. And remember that those receptors can either be located in the cytoplasm or in the cell membrane, depending on whether this ligand is protein-based or lipid-based. Okay, that's going to dictate where its receptor is. And of course, if I ever go too fast, please just pause me. And remember, you can always go back and watch this again if it's confusing to you. Okay, then you have this AP Bio free response from 2013. It says the figure represents a generalized hormone signaling pathway. Briefly explain the role of each numbered step in regulating gene expression. Notice it doesn't say it wants the name of each step. It just wants the explanation of what's going on. So in step 1, okay, here's a signaling molecule. It's binding to a receptor. Okay, step 2, trying to get the message from where the signaling molecule bound to its receptor to the nucleus. Okay, we know that this is called signal transduction, but just stating that this is signal transduction is probably not going to get you any points. You need to explain what's going on here. And then in the nucleus, we're getting the message to the nucleus. So it's probably going to be a protein binding to a section of DNA to let that section of DNA know that it's about to be transcribed. Okay, we're not going to do that yet. So I think you might have the scoring guidelines for this question on your sheet. That's not for you to memorize or anything. It's just to kind of show you what kinds of things the AP exam is asking. They're going to ask you to apply your knowledge, not necessarily just regurgitate. Okay, let's go to the end of notes. All right, you have a sheet that says how insulin works. So this section is entitled When Cell-to-Cell Communication Goes Wrong. And we'll see other examples of this in this unit. So a couple things about insulin that you need to know. Insulin is produced by beta cells in your pancreas, so pancreatic beta cells. If you remember talking about long distance signaling the other day, we said that in order for it to be long distance signaling, you would be putting a hormone into the bloodstream and it would be traveling to a different place in your body to get to its target organ. Okay, insulin binds to receptors on all the cells of your body except your brain and red blood cells, because what insulin does, insulin is like me putting the key in the door. It is needed to open up glucose channels so that glucose can go from your bloodstream into your cells. Okay, it opens the glucose channels. If insulin is not there, the glucose channels don't open and you have too much glucose in your bloodstream, resulting in high blood sugar. So without insulin, too much glucose will stay in your bloodstream and excess water and glucose are excreted in your urine. Okay, those are some of the peeing a lot and having sugar in your urine are some of the diagnostic symptoms of diabetes. Okay, so that's explaining the importance of insulin. It acts like a key in the door. If I open my door, I would be like the insulin. You guys coming into the room would be like glucose, and my classroom would be like a cell. I'm opening the door so that glucose can come into my cell. Okay, next we're going to look at type 1 and type 2 diabetes. And again, I'm trying to get this done quickly before the bell rings. So if I go too fast, please just rewind or pause. Type 1 diabetes is an autoimmune disorder. Typically, this is not diagnosed until about anywhere between age 7 and 10. And it involves your immune cells attacking the beta cells of your pancreas. Okay, it actually ends up destroying them. It doesn't recognize your beta cells of your pancreas as belonging to you, so it attacks and destroys them. So that's what the second bullet says here. The immune system destroys the beta cells of your pancreas that produce insulin. This typically doesn't happen right away. That's why you're not typically diagnosed with this at birth, but it usually happens in your about age 7 to 10. Okay, notice insulin receptors are still present on target cells. Okay, so the receptors are still there, but your body's not making insulin that can bind to it. Okay, so that is the problem with the cell communication. That's the big thing with this, is I want you to be able to tell me, okay, in type 1 diabetes, this is the problem with cell communication. There's receptors, but there's no insulin to bind to it. And then the other part of the real part of the recognition problem here is that your immune system cells do not recognize that your beta cells belong to your body. So they destroy them. So cell communication, just like when you have a communication problem with one of your friends, this is all about where communication breaks down. So immune cells are destroying the beta cells of your pancreas because they don't recognize that those belong in your body. So insulin receptors are still present on your target cells, but there's no insulin to bind to it. So your treatment is injections of insulin daily. Let's look at this side. So in type 2 diabetes, your body still makes insulin, but because of your diet typically, your body has worn its insulin receptors out. You've utilized them too much, and so they kind of get tired and they don't recognize the insulin anymore and they don't bind to it. That is the communication problem in type 2 diabetes. Your body still makes the insulin, but the receptors are basically worn out and they no longer recognize the insulin. So not having enough insulin is not the problem with type 2 diabetes. Obesity and lack of exercise often contribute. And you may take some medication for this, but typically this can be controlled with diet and exercise. All righty.